I am curious about how we can stop brain cancer. I'm Sheila Singh, and I'm a professor of neuro-oncology and neurosurgery at King's College London. And I'm the joint head of the School of Cancer with the School of Pharmaceutical Sciences and the director of the Comprehensive Cancer Center. And I also remain a part-time professor of surgery and biochemistry at McMaster University.
And have had friends with cases of glioblastoma in their lives, and it is a very horrible and aggressive cancer. And so it was great to see the research article. I'd love if you can tell me more about that.
So, together with colleagues at the Hospital for Sick Children in Toronto, we have been working long and hard to try to understand how glioblastoma is able to invade the brain. Because you are right, it is a deadly and lethal tumor. And part of why it is so difficult to treat is because it's not one discrete mass. There are other brain tumors that we treat that are discrete masses that don't invade into the brain. And you can imagine, even for a neurosurgeon, it's inherently easier to treat something that is like a nice tennis ball sitting in the brain that you can carefully dissect around and remove in its entirety, as opposed to a glioblastoma, which is more of a diffuse mass that has tentacles that spread into the brain. And in fact, it has a way to infiltrate the brain and send tumor cells into areas that are no longer operable. So most neurosurgeons will tell you, I did a gross total resection of a glioblastoma, but the emphasis is on the word gross. We can only resect what we can see microscopically. We can never surgically treat the invasive tumor that spreads. And moreover, some of the tumor, the glioblastoma may begin in one hemisphere of the brain, but because of its invasive ability, it's actually able to travel across the white matter tracts that bridge the two hemispheres and invade the opposite hemisphere of the brain, such that by the time someone dies of glioblastoma, it's possible that almost all of their entire brain is infiltrated with tumor cells. So this is not a surgically treatable tumor. And then you would say, okay, so how about a treatment like chemotherapy or whole brain radiation, where you can just globally treat all of those cells that are everywhere? But the problem with chemo and radiation is that they only treat actively dividing cells. So in order to be hit by chemotherapy or radiation therapy, you have to be rapidly proliferating. And if a cell is not proliferating, it can evade or escape chemotherapy and radiation. So we just don't have a good treatment yet for glioblastoma. And so I think part of the nature of that is because of how invasive and infiltrative it is. And that's why we decided to focus on, we already have made some headway with surgery, chemo, and radiation. How do we now tackle the part of the tumor that we can't treat, the infiltrative invasive front?
Yes, I can't wait to hear more about that and how you did that. And the way what I read was really fascinating. So please tell me more.
Of course. So, in order to do this, we really wanted to try to understand more about what signals not only the GBM cells generate themselves, what we call cell intrinsic signaling pathways, are things the tumor cells do themselves to regulate their behavior. But we also wanted to speculate that there might be signals coming from the brain itself that are in the brain cells. Within the brain, normal cells could be interacting with the tumor in such a way that it facilitates its activity. And you probably know that there's been a huge explosion of research in two areas. One area is about the tumor microenvironment. So we've begun to realize that it's not just the cell intrinsic pathways that govern how a tumor forms and invades, but it's very important to consider the cells in the environment that could be signaling to the tumor back and forth as well. And the second sort of cool area of research that has been exploding recently is called cancer neuroscience. And cancer neuroscience is this very cool, as you can tell from the name of the field, merging of two fields, whereby we've started to interrogate the crosstalk that happens between normal cells and brain cells. And we've begun to realize that sometimes normal cells can also be a player in terms of dictating how the cancer forms. And so where we started with all of this work is that really we were trying to figure out Are there normal brain cells that interact? Is there an interplay between brain cells? So we went to the literature first, and there's really good papers published by my colleagues Michelle Manji and Frank Winkler talking about how neurons can actually have electrophysiological communications with glioblastoma cells. And then others, such as Manuel Valiante, have published that astrocytes, which are another subset of supportive cells in the brain, can also signal to GBM cells. But there's a third class of brain cells, and they're called oligodendrocytes. And oligodendrocytes are the cells in our brain that usually make myelin. And myelin is the insulation for axons, or those white matter tracts that I spoke about, that are like the electrical circuits that connect all of the cells in the brain. So if you don't have myelin, you don't have insulation. And so what we did in the very beginning is that we actually wanted to try to look at all of the published literature out there in terms of looking at single-cell sequencing maps of glioblastoma, and we interrogated these single-cell maps from primary and recurrent tumors. And what we realized is that there are glioblastoma cells that recruit oligodendrocytes to the border of the tumor. And all of these cell signaling maps told us that what would basically happen is the GBM cells have a way in step number 1 to attract the oligodendrocytes over. So imagine that you have this cell that's not supposed to be there, like a stranger, and it calls over to someone and says, "Hey, you, come here." And the oligodendrocytes are minding their own business, making their myelin, doing their thing, and then they get a signal from the GBM cell. And in this case, the signal is called CX3CL1. And the only significance of that is that it's something called an inflammatory cytokine, or it's like a signaling molecule that you can send out to get someone's attention. And the way you can get someone's attention is if they have the receptor for that. And the oligodendrocytes have the receptor for that cytokine. So step number 1, the GBM cells call the oligodendrocytes over. And then the very interesting thing that happens next is that the oligodendrocytes become, let's say, converted to what the GBM cells are telling them. The GBM cells then say, "Hey, listen, I need your help." And the oligodendrocytes, usually they just make myelin and they're intrigued by this, and they start getting other signals. Now the GBM cells that the oligodendrocytes have been recruited to the tumor border, and they're now directly next to the GBM cells. Now the GBM cells send another signal called interferon, and this changes the fundamental nature of the oligodendrocytes. Before, the oligodendrocytes were cool, calm, making myelin, but now they've gotten this interferon signal and they change into what we call reactive oligodendrocytes. Reactive oligodendrocytes, they're the ones that actually are signaled to when the brain is injured. When the brain is injured, the oligodendrocytes wake up and they start saying, "My God, we have to repair this injury," and they go into this hypervigilant reactive mode. So the GBM cells have now converted the oligodendrocytes into these reactive oligodendrocytes. And now that they're reactive, they're starting to secrete cytokines of their own. And the cytokines that they're secreting are called CCL5. So now these reactive oligodendrocytes are secreting CCL5 back to the GBM cells, and the GBM cells possess that receptor. They have CCR5, which is the receptor for the CCL5 ligand. And now this has empowered the GBM cells to be able to invade and migrate. So basically what the GBM cells have done is they have hijacked the oligodendrocytes. They have converted them from being sort of good citizens to aiding and abetting a criminal, and they're now helping the GBM cells invade to the opposite hemisphere.
Let me just see if I can rephrase it for you. Basically, that there are these glioblastoma cells that are trying to enter the brain, and then they convert the oligodendrocytes, and they convert them to basically help them out in order to spread more widely throughout the brain and to the other hemisphere.
Exactly. So the oligodendrocytes are recruited to help these GBM cells invade. And that happens through these signaling axes. And we saw these signals in the single cell data that we interrogated. And we were wondering, what's this CXCR31 doing here? And so that's how we uncovered this new signaling axis. And we found it in what we call in silico data, because single cell sequencing data is just a bunch of databases published by other people. So we developed experiments in a dish using oligodendrocytes generated from stem cells. So we basically take stem cells and push them into the oligodendrocyte lineage to make oligodendrocytes in a dish. And then we drop glioblastoma cells from our patients at McMaster into a dish. And we actually can now recapitulate what we saw in the single cell data in a dish. And we verified that that signaling that I was telling you about, especially the CCR5, CCL5 signaling, was all happening. And once we saw that happening, logically, our next step was thinking, can we block this? Now on to the next exciting part. We've identified a new signaling axis between a normal brain cell, oligodendrocyte, and a glioblastoma cell in the brain. And this really empowers us now, because now we understand this communication is happening. If we can block it, perhaps we can block the invasion. Fortunately for us, when we looked for drugs that blocked CCR5, there is one already being used for a completely different purpose. And this drug is called Maraviroc, and it's actually used to treat HIV patients. And so we thought, this is phenomenal. There's already an FDA-approved drug that blocks the signaling axis. And this just basically gives us another indication for this drug. We should try this drug in glioblastoma patients. We can't do that right away because it's not approved by the FDA. So I can't quite give it to a glioblastoma patient, but we can model it in this beautiful patient-derived xenograft model we have in the lab. So in the lab, we take immunocompromised mice. We engraft a human patient GBM tumor into those mice, and then we can treat the mice with different experimental therapies. So the mice are our patients in an experimental clinical trials unit. And so we were able to treat the mice with Maraviroc, and we were able to prove that it really does block invasion of the GBM in mice. So this is an excellent proof of concept. This is enough data to convince a drug company or an industry partner to try this now and to file an application to Health Canada or to the FDA to consider the use of Maraviroc in a glioblastoma patient.
And so the clinical trials for this, when will they begin?
So we have to find funding for this. We have to find the right partner. We have to find, a way to get physicians involved who are willing to give Maraviroc to patients. So we still have a little bit of legwork to do. But again, all going through the clinical trials for a brand new drug would probably these days take 5 to 8 years. I think we can probably run a bit faster. So I'll be hopeful and say, as we're starting this process, maybe we're looking 2-3 years down the road.
Are there major side effects for HIV patients?
No, surprisingly, this drug is pretty well tolerated. It doesn't have a terrible side effect profile. And certainly, it doesn't have any worse a drug profile than the drugs already being used to treat GBM patients.
Yeah, hopefully that process can be done quickly and more people can benefit from it.
Yes. So what we've heard from our glioblastoma patient community is that it's both gives them both hope and it's frustrating because as we have colleagues at this wonderful funding agency called Brain Cancer Canada, and they have a lot of patient advocates in their group. And one of them said to the director of Brain Cancer Canada, it's like dangling a carrot in front of a hungry donkey. We want to take the drug. We're not quite there yet. We have to do things through the proper route. And I wish there was a way to expedite the regulatory pathways, but we have to go through all these channels, even for a repurposed drug. I will say that in the US, I really admire the fact that you guys have more pathways to repurpose drugs for patients, more regulatory pathways. And so I think that there's a possibility that we could work with our US colleagues and maybe do this even faster.
Why did you decide to go into this field?
So since I was a child, I've always loved everything to do with the brain. And when I went to medical school, I tried everything with neuro and the word neuro in front of it. So I tried neuropathology. I rotated on neurology, which is the medical counterpart of neurosurgery. But the reason I chose neurosurgery is because it felt like activism, right? It felt like someone came in with a large brain tumor and you would say, "Don't worry, I'm going to fix this. I can take it out and make you feel better." And you can as a neurosurgeon. But what we realize in neurosurgery is a lot of the things we do, while they're amazing, they represent quick fixes that aren't necessarily durable. So I began to realize that no matter how much neurosurgery I did, I would not be able to cure something like a brain tumor unless I did more basic science or bench science.
What do you see in the near future in this area of glioblastoma?
So I think the clear thing with glioblastoma is that it is a very heterogeneous tumor, meaning that there are all kinds of different cells with different signaling pathways communicating with different brain cells. There's just too much going on. And to me, what this means is that a single drug therapy is unlikely to work. So what we really need to work on for glioblastoma is logical and rational drug combinations. And perhaps these drug combinations need to be personalized or tailored to every patient. So what I really hope we start doing for glioblastoma patients is more precision oncology or precision medicine approach where we personalize the therapies to tailor it to the biology of their tumor. I think those tailored combinations will be more successful than just the blanket therapy we're giving now. And the second thing is immunotherapy has had huge successes for blood cancers, what we call liquid cancers, but we haven't progressed as well with solid tumors. And so I'm hoping the next decade will show some successes of new immunotherapies for glioblastoma.
Dr. Sheila Singh, thank you so much for joining me today. It was so wonderful to hear about your research, and I wish you the best of luck in making this happen. It will be so important and helpful for many people.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.